Today we have two excellent lectures. The second one will be presented by Professor Ann-Christin Pöppler from the Institute of Organic Chemistry of the University of Würzburg, Germany. And the title of her presentation is Molecular Level Structural Information through NMR Spectroscopy and Complementary Tools from Bridging Solid State and Solution to Detailed Structure Property relations.
Before Ann-Christin starts, let me introduce her. She, since 2016, is an independent junior professor for organic structural chemistry at the Institute of Organic Chemistry, University of Würzburg, Germany. Her long-term research interest is development of new, improved analytical approaches in the context of drug delivery. Professor Pöppler aims to achieve this through detailed molecular level understanding and identification of essential structure property relations, combining NMR spectroscopy with synthesis and with variety of complementary analytical methods. Ann-Christin, since 2005 up to 2010, studied chemistry at the University of Göttingen, Germany, and next she was doing a PhD under supervision of Professor Starke, and in 2013 defended with distinction her PhD thesis entitled Advanced Methodology for Investigation of Organic Compounds in Solution. Then she worked for a year as Dietmar's research associate and moved in 2014 to the Max Planck Institute of Biochemical Chemistry, Göttingen, where she got a postdoctoral position in the group of Professor Adam Lange. Next, in 2014, she moved to University of Warwick, UK, where she worked for 2 years as a postdoc under supervision of Professor Stephen Brown. Ann-Christin got several prizes and different awards, such as, for example, the Felix Bloch Lectureship of the Division Magnetic Resonance by the German Chemical Society in 2021, and the Fulbright Cultural Award of the German-American Fulbright Commission in 2020. She also got several teaching awards and different German and British grants and fellowships, including Fedor Lyon Research Fellowship for postdoctoral researchers, Newton International Fellowship of the Royal Society, and funding from Royal Society and research grant from DFG. Dear Ann-Christin, the screen is yours.
Thank you so much. Thanks for the very, very kind introduction, and also for this great possibility of having this tandem talk with Professor Starke. As you have already said, it's for me quite a special occasion to give a talk together with my PhD supervisor. And it's also a special occasion because it's quite out of the general community with who I interact. And this is the thing that I particularly like about today, because I would really love to come into contact, to start discussing with you and to exchange ideas, because I think that's something really important to science, that we have the possibility of crossing boundaries, not just countries, but also subjects, and that we can work together to advance the things that we are interested in. And today I would like to take you on a journey. Most of this journey is actually covering how to bridge the solid state in solution. So picking up from what Professor Starke was just talking about. Thank you for the really good start that I can have and the good connectivity. And then I will advance from there to go also to structure property relations and see how we can combine different methods to actually learn more of the things that we study and correlate the individual properties with the specific functional moieties. And this is also something that, as you mentioned, that I'm really keen on teaching, and this is something that I also try to teach the students during the courses, that there's not just one tool that fits all the problems, but that we often need complementary analytical approaches and other approaches, also including synthesis, so that by combining all of them, we can actually get the best out of the question or complete the puzzle that we have of information. So this is something that is very close to my heart and very important to me with respect to the research. And what is also very important to me is that in general, nobody of us is doing what we're doing alone. So I like to compare it with this image that our job is like a bank robbery, and without the right accomplices, it will not work. And this is why I need to thank most of all to my team. So since 2016, they are really supportive. I have the pleasure of working with really motivated and generous human beings, and it's really fun coming to work every day. You can see here their office window, and of course this also is really important with respect to the collaborators, because they always come up with really exciting questions and problems to solve, so that it's an absolute pleasure to be working in this area. And as already mentioned in the kind introduction, I also wanted to mention the different stages and wanted in this moment say thank you to Professor Starke and also to Michael John, who actually gave me the starting point to have a look at this, to start with looking how we can translate things or bridge things from the solid state and solution. And without their cool offer for this topic, I probably wouldn't work in science anymore. So thank you for this opportunity. So I would like to start with citing something from the International Union of Crystallography, because since 2014 there's a new commission on NMR crystallography and related methods. And NMR crystallography in this context is defined as the study of solid and or crystalline materials by NMR spectroscopy. And with this slide, I would like to stress the complementarity between NMR, which is generally a short-range technique, and then X-ray, which is more a long-range technique with respect to the order phenomena that you're looking at. And in particular, I would like to present you today some work that will involve all the three, so combining those two also with quantum chemical calculations, and that sometimes it's not possible to interpret the NMR data, for example, without the help of a tandem of the other two. And at this moment, I would also like to thank Professor Wozniak for the kind invitation to his seminar series. And I would also like to include some thoughts from one of his papers from the 90s. And he didn't just do the mixture of the techniques, but he actually did also crystallography or NMR on individual single crystals, which is really fascinating and something that I didn't have the chance of measuring yet. And what I really liked from this study, hydrogen bonding and structure of substituted quinoxalines. And what is written here quite in the beginning of an introduction, you can see it highlighted, is that the study of intermolecular interactions is often the aim for both the NMR spectroscopist and the crystallographer. So this means that in the end, we are all interested in more or less the same thing. We're just looking at it from different angles and or with different eyes, which is something that is really fascinating and could be exploited even more than it is been done. So with this, I would like to shift more towards the science, and I would like to connect to what Professor Starke was talking about in his talk during the last 45 minutes to an hour. And in this case, I will start with organometallics, connecting to his work, but I will not stay with organometallics. So the focus in the presentation today will not be on the specific class of compounds, but more on the complementarity and how we can bring together the different techniques. But this part here will be on organometallics and bridging the solid state and solution. So this is always indicated by this bridging symbol. And it all comes down to, if we do chemical reactions, they are most often done in solution. And the question is, how do we know which the reactive species is? What is present in solution and what is the reactive species? And normally in organolithium chemistry, as Professor Starke already said, it's very air and moisture sensitive. So generally the reactions look like this with extensive Schlenk techniques. And there are also several people who are involved in trying to find out what is happening on a molecular level. One of them being Dave Collum in the US, who I also had the pleasure of meeting on a conference in Germany a couple of years ago. And there's some very recent work from his group where he studied a sodium component, so not a lithium, but we are now with sodium. So instead of lithium HMDS, he has sodium HMDS. And there's one crystal structure in the solid state, and then this crystalline arrangement was broken up by a set of several solvents, so different natured solvents, and then they found an incredible amount of structures that formed upon dissolution of these solid materials with the different solvents. And this is just to give you an idea that even though we have quite a clear image of what is happening in the solid state, we cannot rely on that it will always remain the same once we switch the phase and we go into solution. And I would like to show you this on an example from the time with Professor Starke, which was actually the first manuscript or the first full paper that we published together, and it was also the very first time that I got into contact with solid state NMR, even though it was still as a, let's say, service unit at the Max Planck, but this was like the first contact also with what I'm mostly doing now. And the structure that I'm showing here was synthesized and also crystallized by Maggie Meinholz. She was also a PhD student in the same group, and she crystallized this really beautiful co-crystal, which consists of one tetramer of tert-butyl lithium and one tetramer of a lithiated dimethyl aniline. And you can see here the two tetramer clusters. And the first question was, is this one single crystal, or is it actually the bulk material? So is everything co-crystallized, or is only the crystal that was picked co-crystalline? And then you can check multiple crystals, which of course she did, but you can also measure NMR of the powder, and of course you could also do powder X-ray diffraction then in an inert atmosphere. And in this case, both the multiple diffraction experiments and also solid state NMR showed that this is indeed the bulk material, so that all the compound that was obtained is this co-crystal. And then the second question was, so how does it behave now if we put it in solution? So if we dissolve this crystalline material, then you can see here 7 lithium NMR data. And you would expect from this initial crystal structure that you would actually have 2 signals, one for the right lithium cluster and one for the left lithium cluster. But in the end, what we found back then was that we had 5 different signals, and that these 5 different signals also diffuse differently. So the right side, you can see like a chromatographic evaluation of these mixture of compounds, and you can see that there is a regular shift in the diffusion speed that you can see. And what we found here by these 2 experiments is that overall in solution, we have an interconversion of these initial dimethyl aniline lithiated version. We have a 3-to-1, a 2-to-2, and 1-to-3, and then the complete tert-butyl lithium. So you can see that all different mixtures are actually existing in solution, while in the solid state, there's only this most stable form. And this was an indication that we have to be careful if we cross phase barriers, like in this case, going from the solid state to solution, that the structural information is not necessarily retained, but that it can actually happen that we have something different existing in solution than what we have as a solid state structure. And this was a really encouraging study, which showed how the different techniques can complement each other. And this complementation is also something that we tried much later. So this is a very recent work, and we are now completely in the solid phase. So that was the question, if we are in a solid phase and we want to characterize something in the solid phase, of which we do have crystal structures, is this possible, and can we always do this? And the molecule that I have picked here as an example is paclitaxel. It's a natural product, and it's very important in the fight against breast and ovarian cancer. So it's one of the most common drugs that is used in the treatment. You can see that it is rather complex, so it has many functional groups. It has 47 carbons. more than 50 protons, nitrogen and oxygen. And if we measure solid state NMR of such a compound, we can get quite a beautiful spectrum, which you can see here in the bottom. So for the current situation, it's just enough to see that there are lots of spikes, and that there was a partial assignment of these peaks available. So as an NMR spectroscopist, if we want to look into the substances and their arrangement in detail, we of course want to know every peak and which functional group the peak belongs to. And in this case, we had some help from a previous literature. However, it was not a full assignment, and we were facing one problem. And for this problem, please focus on this molecular unit highlighted in red. This is a ketone functionality, and it's the only ketone that is present in the paclitaxel molecule. And in red in the spectrum here, you see the highlighted position where the signals come, and you see that there's not just one signal for one molecule, but there are actually two signals. And these two signals are directly giving us access to crystallographic information, because the number of signal sets in the solid state NMR spectrum corresponds to the number of independent molecules in the asymmetric unit of the crystal structure. So we can immediately say that we have a Z prime equals 2 structure of this crystalline material as we bought it. So this is the as received spectrum. And this means also now that from the NMR point of view, how do we know if the left or the right signal belongs to molecule A or molecule B? And how do we know this for all the other signals, which are also there in duplicates? And this is when we wanted to combine the different things, when we wanted to get the help from both crystallography and from calculations. And we checked the literature. There were 3 different structures from the Dinnebier group in Stuttgart of paclitaxel without any organic solvents. So these are now dihydrate, hemihydrate, and anhydrate structures. And what you can see from these crystal structures from the database is that the hemihydrate and the anhydrate were obtained in situ using powder diffraction, while the dihydrate was obtained by single crystal synchrotron diffraction. So this would be the structure with the highest quality. And it is important that there are these in situ structures, as we will see later. So this means we have available crystal structures on this molecule. We got it, purchased the molecule as it is. And in the last decades, there was a massive advancement in calculating NMR parameters, in particular of periodic structures. And this meant that we could set out and calculate for such a given crystal structure what would be the expected chemical shifts, and then we could use this for assignment. The problem is that these calculations turned out to be more time consuming than we were hoping for. So the program package that we used was CASTEP. You can see here the unit cell, which is the input for the geometry optimization, which had almost 1,000 atoms, and we had quite a big bunch of memory available on our local cluster, but still just geometry optimizing the structure took us 2 months. And this was just for this dihydrate with which we started, because it was the best defined structure. And you can imagine how really happy the PhD student was when he finally got the fully optimized structure, could do the NMR calculation, and then plotted the experimental chemical shift against the calculated ones. And this is what you see now on this slide. So in top in black, you see the experimental NMR spectrum. And in the bottom, you see the calculated spectrum for the dihydrate structure as we obtained it from the CCDC, then geometry optimized it and calculated the stick spectrum. And you can see, if you look and compare the two, that it's definitely the same chemical entity. So there are lots of similarities, but in particular, if we look into this red area, you can see that our calculations are quite far away from what we have in our experiment with respect to the chemical shift. And the accuracy for organic normal molecules that you can obtain is normally 2 ppm in carbons. And we are far, far away from these 2 ppm difference between experiment and simulation. So a bit frustrated by this, the PhD student also calculated the hemihydrate, and he also calculated the anhydrate, which I'm not showing you here, but shows the same features. So still, there are similarities, like here in this area, but there are vast differences in this area highlighted in red. So something was not correct here. And in the end, serendipity helped us because we had to measure or buy another batch of the same compound. And we did powder X-ray of this new batch, which is the red diffractogram. It's not the most beautiful one, but you can clearly see compared to the green simulated patterns, which are from the database, you can see that there's one additional signal here in the center, which is not present in any of the previously known structures. So either this is a new phase or it is a mixture of phases. And we had the hypothesis that it's a mixture, and we carefully dried this batch too. And then by drying it, we obtained the signal in the middle as the main species, so we could reduce these outer known ones and preferentially get more compound of this inner one. And we then had a lot of help from crystallographers here in Würzburg. So we worked together with a group of Professor Müller-Buschbaum. He's now based at the University of Gießen. And they helped us by measuring in a capillary a really long experiment of this new batch of dried paclitaxel. They did indexing of the cell and also a poly refinement. And they gave us a unit cell that has shrunk by 6.2%. Upon this drying process compared to the crystal structures that were available in the literature. And then we started to predict the structure. So we used Material Studio to simulate the structure. And what we found when we compared the different data is that this new crystallographic phase that is not in the database is not really new, but it's a relaxed version of the anhydride that was initially published by Dinnebier. Because what he did is that he used single crystals, which included water, and heated them in the powder X-ray. But then he immediately measured. And we found through several experiments that if we took the hydrous material and just heated it, we got a material that was partially crystalline and partially amorphous. And then if we left it over time to relax, really also at slightly elevated temperatures, then we formed this anhydride structure. While in the initial publication, the structure is retained upon heating and just the water is leaving, but there was no time left for the crystallographic rearrangement of the structure. And this means that we could make a new structural model, also included in CASTEP, and we are currently completing the solid state assignment for this paclitaxel molecule, so that hopefully in a few weeks we will be finished with this and will have fully understood what is going on in the solid phase, not even changing within the phase. We still had trouble just understanding what is within one solid phase. And this means that I have taken you already through this bridging solid and solution, and we have discussed a bit what is going on for individual crystalline phases in the solid state. And I would like to invite you now to check more what we can chase with the help of NMR spectroscopy, and always what we can do if we complement it with other tools, which is the important part here. And the two things remaining here are crystallinity in a polymer matrix and drug polymer formulations. And this is also why you have this stomach image, because we are working on systems that are meant to be either below the skin or taken orally as drugs. And this is what we will look at now in the remaining time. So the first thing that I would like to present you are how you can study crystalline solids in a polymer matrix. I guess that every one of you is aware of these crystalline sponges that were invented by the group of Fujita, and that you can study a lot of really small quantities of materials by X-ray diffraction in these sponges. And here the sponge is not something that is ordered itself, but here our sponge is kind of a polymer matrix, and this polymer matrix is by nature amorphous, so it doesn't show any Bragg diffractions in the powder X-ray, but we have crystalline solids within this matrix. And all of these crystalline solids, which we have been studying, were in the area of HIV drugs. And here you can see statistics that there are so many millions of people living with HIV and also receiving treatment for HIV. And this means that all these patients have a lifelong, the lifelong need to take daily drugs, so lots of drug products. And fortunately, there are also possibilities now, so-called PrEP, where you can have preventive drugs that will allow that you prevent getting infected with HIV. So there's a lot of things going on, but there's fortunately also research in the area of ultra-long implants, because then there would be the advantage if you had an implant that you could dispose underneath the skin. You would have the advantage that the person doesn't need to take a drug or multiple drugs every day, but that you can actually have a depot underneath the skin. And this is by biodegradation, is releasing step by step the correct amount of a drug. And then it also would have the advantage that if you, for example, don't tolerate something well, it could still be removed. And this is something that we got interested in, these ultra-long biodegradable drug delivery systems. And we studied a molecule called dolutegravir, which is an antiviral drug, and is based on this study, which in 2019 was published in Nature Communications. And you can see here that they did a lot of imaging on these systems. So you can see here in pictures of these ultra-long acting implants. And then you can also already see here indicated, and this is what actually made me interested, is that you can see here crystalline particles. So these things look like you have small crystals inside of this polymer matrix. And we thought that this would be something that is really good or well to be studied by NMR spectroscopy. So we took the systems that they studied here in this 2019 paper. You can see here the polymer is actually a polylactic acid copolymer with glycolic acid. So PLGA is the system. Then we have the sodium salt of dolutegravir, as you can see it here. And this is all prepared in a buffer mixture, simulating the pH conditions that would be underneath our skin. And you can see here that you can make a mixture of the polymer and the drug. And if you drop this mixture inside of this buffer mixture, so this aqueous buffer mixture, you get these precipitation of these polymer matrix systems. And then you can dry them, as you can see here, and can study them with different methods. And we did not just NMR spectroscopy in this case, but also powder X-ray diffraction. And we were studying these solid particles after various times that they were underneath the skin. In our situation, that they were in the buffer solution. And this means that we can watch how they are degrading, and we can also watch how they are releasing the drug that is encapsulated inside. And here you can see the solid state NMR. And what is interesting, so there's in yellow, the pure dolutegravir, so the pure drug. Then we have spectra after 6 hours, 1 day, 3 days, 7 days. And for comparison, the polymer spectrum in the bottom. You don't need to check out the whole spectrum. What I would like to point out is that in some cases, like here signal A, we have a signal that is vanishing or maybe shifting. Then in the second dark gray column, we see that there's a new signal emerging. And what is also curious here is the signal I. If you check out, you have 2 signals in the pure drug, but we have only 1 signal after 7 days. So we can clearly say that something is going on the molecular level. We have shifts, so changes in chemical shift, and we also have changes in the number of signals that are observed. And this is indicating something about the symmetry. Again, like this number of signals is corresponding to the symmetry in the crystal. And powder X-ray diffraction told us even more things. So we could distinguish by powder X-ray diffraction that the unit cell of our crystalline material, so inside we could detect the crystals inside of the amorphous polymer matrix. And so these signals change. So we have a change in the unit cell, and also we have some signals disappearing over time. And overall, we can combine many things from diffraction, NMR, and also calculations and what we learned, and gain a better understanding of what's going on in these systems. So these were the initial images, and solid-state NMR showed that there was absolutely no intermolecular interactions between the drug and the matrix. So this means that we have such a dispersed system. So we have crystals which are dispersed in the polymer matrix, but they are not homogeneously mixed, or they are not also amorphous, but we actually have chunks or domains of crystals and then domains of polymer material in agreement with these initial images. What we could also see is from solid state NMR and powder X-ray that we have a change in the crystalline form. And additional information from the literature is if this polymer PLGA is degrading, we get acidic compounds on a local scale, which means that locally in these systems here, we have a change or a drop in pH, which means that at some local parts where the degradation is taking place, our pH is lowered considerably, even though the whole solution is in a buffer. And this helped us to explain that what's happening here is that the sodium dolutegravir that was there initially and that had this dimer formation in a crystal structure, so two sets of signals, was protonated because of these local high concentrations of H+ ions, and the sodium was released from the structure into the solution, and this was then the change that we observed both in the crystalline form by solid-state NMR and by powder X-ray diffraction, and we could also confirm this because there are patterns for this form which also show the same powder X-ray diffraction pattern. And this leaves me to go to the real amorphous situations. So this is where we would have the strength of NMR, that we don't rely on long-range order if we are interested in this short range or what's happening on the short range. And in particular, I would like to show you what you can learn using polymer formulations. And again, this is not just using NMR, but it's always complemented by calculations. Otherwise, we would have no chance of analyzing these systems. So what we study are tri-block copolymer micelles. So you have here a copolymer, which means it consists of different monomeric units, and the blue ones are indicating more hydrophilic environments, and the red ones more hydrophobic environments. And if we have all these polymer strands in solution and they surpass this critical micelle concentration, they self-assemble in aqueous solution into these nanometer-sized objects. So these nanoparticles are said to have a core shell structure with a fairly, or ideally, a well-defined distribution, with the hydrophilic parts being on the outside, so the shell of this structure, and also providing solubility, protecting this particle, while the inside consists of the more hydrophobic elements, and you can transport also hydrophobic materials in the center. But as you will later see, this is not as straightforward as you would think. But now I have to go back to this bridging thing that we started off with. So if we have the strict definition of micelles, then a micelle is something that is swimming around in solution. And if we have polymer and a drug, here this is another HIV drug, and dissolve everything in chloroform, so we break up any aggregate that you can find, then you can see that you have NMR signals for all the individual components. The thing is, if we go to aqueous solution where we would use them, give them to patients, whatever, in this area here, in particular the blue one, we don't have any signals. So from our solution data, we cannot tell anything about what's going on in the center. And this is the reason for this is that the particles are so dense and so tightly packed that they behave almost like a solid, and we don't have access through solution NMR or other solution techniques to get insights into this core. And this is why we started to do a lot of solid-state NMR on these systems. But I have to ask in this moment, is it even allowed? I mean, we have seen all these situations where we have a nice crystal structure, we dissolve it, and it turns to something completely different. And now I'm telling you that I have these nice micelles swimming around in a solution, and I will freeze dry them. And then is the question, do I still have the same, or do I have something completely different? And one other idea to bridge solid state and solution is, I mean, what I could do is I could just freeze dry it and redissolve it and check that it's still intact. But this is kind of very black box and very indirect. So what I would really like to do is I would like to study the micelles in a solid form while they are still in the same environment like they were before. And the way that my PhD student Sebastian came up with was to fill the solution inside of a rotor, cool it down to really low temperatures, and then measure ice cubes in our NMR spectrometer. And then we are bridging again the solution with the solid state, just the other way around like we did it initially. And he did this for glycolipids in this case, and you can see here a comparison between a freeze-dried glycolipid and a frozen solution of a glycolipid, and he found that the data that we obtain resemble each other very strongly. There are small subtle differences also in the signal to noise, but generally we can compare them. And this was very encouraging. We are, of course, performing this for lots of other systems now because it's a very curious way of bridging solid state and solution, and we are quite excited about this possibility, and let's see what will come out from this in the future. And this means we can at least deduce some things from our solid state measurements for the situation in solution. And I will only briefly show you what we did with Professor Luxenhofer from Helsinki, who synthesizes these polymers, and he encapsulates the natural product curcumin inside of the polymers. And he and his group, they are experts for a decade now in this area, and they are capable of doing something really cool. They can encapsulate a large amount of a guest molecule inside of the polymer, inside of micelles. So we have something between 17 and more than 50 weight%, and this means that we can study really what the differences in loading are doing to the structure of the micelle. So we have 3 different forms: low, medium, and high loading. And you can see here on the right that in the colored diffraction patterns, that all of them are X-ray amorphous. So we cannot get detailed information like for the crystalline material. And this means we can study these amorphous systems, but with the question in the back of our mind, why is it possible that they can load up to more than 50 weight% of such additional compounds. And we also check properties, because we are now in the section of structure property relations, and you can see that the dissolution is good for the low loading, it's okay-ish for the medium loading, and it's really crap for the high loading. So we have to compromise the dissolution, and again the question is why? Do we find something on the molecular level to explain this? So we did lots of solid state NMR experiments. I will not go into detail of the spectra. You can see here that they all differ. Doesn't matter whether you have individual components, the formulations, or amorphous materials. So we see differences, which means we can interpret differences. And I would like to focus on what, from the complementarity of the methods, we could learn. And again, you can see here the CASTEP symbol. So this was heavily helped by calculations. And what we found just from NMR was that if we load more and more of this compound inside of the polymer, it gets less ordered. So we have no crystallization nuclei starting to progress or forming domains like in the previous example. With calculation and NMR, we found that these polar groups on the outside are the main hydrogen bonding partners, and that this enol is happy by itself. And we found also that while in the crystal structure the molecules are twisted, like in this green example, inside of the micelles they are more planar. So this twist is less strong. And the most curious thing in the study was that while this molecule is fully soluble and we would expect it to be in this red part of our particle, we also saw changes for these hydrophilic parts. And this helped us. Well, at first it was surprising, but it helped us to explain all the properties, because if you load more and more molecules inside of the core of this particle, at first you have lots of coordination sites available. They become more and more saturated. And when you have all the coordination sites in the center used up, then you will need the hydrophilic parts of the polymer to stabilize the particle. And this means you have a competition now of curcumin as a guest molecule with the hydrophilic parts and water with the hydrophilic parts. And this means if water cannot approach these arms anymore, then it cannot provide solubility, or the arms cannot provide solubility. And this is how we can explain that the increase in loading is making at the same time a decrease in the dissolution rate. And this leaves us to consider whether we have possibilities that we can alter the polymer structure so that in the end we might get something that has a high amount of gas and a high solubility, and this is something that we're currently testing and working on. And as a last thought, that what I have shown you here on this slide is very schematic. So I have still drawn a pretty picture, but it's nothing on the molecular level. It's not like what you get if you get a nice crystal structure and you really have a picture of your molecule. So we used evavirons as a toolbox molecule, and evavirons for an NMR spectroscopist is like this massive playground, so we can measure many, many spectra. And this is also what we did. So we measured proton-carbon correlations, and then we can see all the colored parts. So gray means carbon is close to proton, backbone, and aromatic ring. Then we can measure fluorine carbon, and we can see the fluorine-containing group is near this butyl chain in the polymer. We can measure nitrogen and proton and detect the blue proximity, and we can measure proton-proton, and we can detect the red proximities. So you can see already that we can start of kind of envisaging where this molecule could sit in the pocket of our polymer, but it's still far away from an actual representation. And this is why we approached Professor Sotriffer from the pharmacy department, and he is an expert on molecular dynamics of proteins and protein binding pockets. And if we have an amorphous system, it means we have a distribution of similar but not identical environments. And maybe we can represent these disordered systems by also getting a distribution of environments from molecular dynamics. You can see here the first attempt of simulating the whole micelle. And then what we did from this simulation is we wanted to extract individual parts and countercheck them. We found the closest distance also to be between these two groups, so in agreement with our NMR measurements. But currently we are at the stage where we need to take snapshots from the MD and calculate NMR parameters, and this is taking a lot of time, and we need to come up with better solutions for that. So I'm happy for any suggestions. And this leaves me to summarize. So we have had some projects on bridging solid state and solution. And we had first the co-crystal and also the ice measurements. I could show you some structural property relations in crystalline samples of paclitaxel and these ultra-long delivery systems. And in the end, we looked at amorphous samples. And what we are currently trying to do is do collaborations with groups involved in machine learning so that we can maybe advance in this MD direction. And with that, I have to thank you very, very much for your kind attention. And I'm really looking forward to your questions and to the discussions. And thanks again to the organizers for this really, really great opportunity.
Thank you very much, Ann-Christin, for a really beautiful lecture and for sharing with us your excellent, very interesting results. And I hope there will be some questions soon. Please put them down into chat. But before this, I will take this opportunity to comment or to ask my question before the first ones will appear on chat. Do you think that different forms of ice, when you freeze these molecules in ice cubes, do they influence conformation and properties of your micelles? I mean, you can try with ordinary ice, or I mean, in total, in general, there are 20 different ices. But you can also try in amorphous ice. Hey, people who are doing electron microscopy, that's very easy. Have you tried to perform this kind of experiment?
That's a great question. So the answer is yes, but we've only started. So what we usually try is that the ice is amorphous. So we usually fill the sample and then immediately put it into liquid nitrogen to make sure that it's amorphous and it's not let's say, by the crystallization process influencing it. But we recently started to also do slow cooling to have ice crystals in the fridge overnight and then measure these ice crystals. The problem is that you have to be really quick so that they will not melt on the way to the spectrometer. So even if we have something at 245 kelvins, then take it out of the spectrometer and open the lid, it already starts again to melt. So this is a bit the danger, but we are working on this, and we are also, in comparison to what you said about the cryo experiments, we are also trying to check if the addition of cryoprotectant to samples is making a difference. So there's a lot of things that, or let's direction that we are trying, and it's really, yeah, it's a curious thing, and I really like it.
Mm-hmm. Thank you very much.
And Dietmar is asking, making ice cubes inside the rotor, would the rotor survive the stress? Isn't the lid coming off sometimes?
Yeah, so if you would fill it to the top, the lid would certainly come out. Fortunately, it already does so during the freezing already in the liquid nitrogen, not inside of the spectrometer. However, the PhD student is sweating like hell whenever he's performing the experiment. He's really scared of damaging anything. So with what they usually do is that they leave a gap below the cap, and then they make sure that it doesn't have any chance to melt. And we rotate it up to 10 kilohertz usually, so we are not going to the maximum, so that the frictional heating is not influencing what is happening in the rotor.
Before next questions will appear, you mentioned at the beginning this, I would say, joint use of, say, single crystallography and NMR. There could be, I remember from that 90s, that there could be some very malicious cases, especially when you have symmetric moiety. For example, two symmetric moieties in the independent part of the unit cell at special positions, which are giving you, for example, two halves, which are giving you formally one independent molecule, versus one molecule in the independent part of the unit cell in general position. So NMR is not able to differentiate those two cases, and really we had a few of them in the past, but that's just a comment.
No, that's very treacherous. Yeah.
Another point: why not to calculate NMR parameters from detailed, say, X-ray studies?
There are actually groups there's one group in Lausanne, and they are calculating ORTEPs based on the NMR data. So there are multiple attempts of, let's say, crossing the boundaries, but it's still very sparse. And they were also in these, let's say, earlier days of the calculations. Initially, they used the X-ray structures as they are, and then calculated the NMR parameters and found that they are quite far off. And then they started with the geometry optimization to counterbalance the distances between protons and heteronuclei. And then they also tried neutron structures, and they calculated the NMR parameters based on neutron structures, and they fitted fairly well even without the geometry optimization. So there was a lot of work, let's say, in the early stages going on with that. And there are also groups which are using individual molecules, creating clusters of like 9 to 12 molecules surrounding one molecule, imitating different crystal structures, and then calculating the NMR parameters. So there are multiple approaches parallel.
Yeah, and these days with Hirschfeld atom refinement, we have even better tools to look for this correlation between properties of hydrogen bonds and NMR parameters. So that could be a great project to find like nice correlation between very accurate and precise distances obtained either from Dietmar's periodic Hirschfeld atom refinement or from ours, which is using clusters. So, and correlate this with NMR results.
The only challenge in that respect is that generally, if you record these really high quality X-ray data, you're recording it at very low temperatures, while the experiments that I'm doing, even if I apply cooling, are way above that. So this would be something that would needed to be taken into consideration when comparing the two.
This doesn't have to be very high quality, or well, high resolution. Quality is also still good.
Yeah.
High resolution data, so you can apply this better scattering factors to room temperature data.
Yeah.
It's enough to have them normal resolution.
Okay.
And these days, of course, there is yet another excellent method, or methods rather, which one could include into this portfolio, complementary techniques. I think about electron diffraction and electron microscopy.
Yes.
I mean, this used together with solid state NMR, with crystallography, it's almost all problems in chemistry could be attacked with this kind of selection of methods.
The cryo electron and microscopy and also diffraction, they yield really beautiful things. Yeah.
Yeah. So you mentioned this powder results. They could be somehow, say, supported by this electron diffraction or cryo.
There is no more questions.
Comments?
Yeah, if there are any questions afterwards, then I'm always happy for emails or other ways of starting a contact.
There is one. Paulina, please read it.
Yeah. Ann-Christin, is there a chance to orient the crystal in the magnet?
In that case, this is something that your colleague has already been doing.
Yeah, that's single crystal NMR. Right?
Single crystal NMR. Yes, that's like another dimension of NMR. So
have you ever heard about crystallization under the magnetic field, which would force orienting?
Yeah, both. I mean, there's the possibility of, for example, if you have liquid crystal materials, then they orient in the magnet when they crystallize. But there's also the group in Cardiff, and they are doing crystallization process within the NMR machine, and then they record one spectrum of a solution and one spectrum of a solid, and they alternate it, and then they calculate how the different crystallization processes and which, let's say, metastable phases are. Yeah, this is really cool work. It's the Cardiff people around Cologne huge, so that's really cool.
Mm-hmm. Ann-Christin, have you ever come some cases of influence of Coriolis forces on properties of your systems? I mean, dependence on the speed of rotation.
Yeah, so we have currently a manuscript under revision, which is called "You Cannot Fight the Pressure," because we have 4 or 5 compounds, both crystalline and amorphous, and they had phase transitions upon magic angle spinning. So this gave us quite some trouble. Both, we even had a crystalline to crystalline phase transition. Yeah, so this is tough, and the only thing you can do is spin at lower frequency, and then you will get more ugly spectra. So yeah.
Yeah, excellent. Okay, we have some congratulations for excellent lectures from Uli Englert, from Carlos Alberto Andrade Serra dos Santos. So we also congratulate you both for these excellent lectures, and thank you very much.